Bonjour à tous. Donc, le seul avantage d'être président du jury, c'est que tout le monde est obligé de m'attendre. Donc un peu en retard, je suis désolé. Voilà. Donc je vais introduire les membres de ce jury. Même si je suis un peu en dehors du domaine, j'ai le plaisir de tous les avoir côtoyés ou de les connaître de près ou de loin. Donc tout d'abord, nos deux rapporteurs venus de Suisse. Donc le professeur Catherine Graham au VSL à Zurich. Et puis Daniele Silvestro, qui seront les deux premiers à prendre la parole. Et donc normalement les plus critiques par rapport au travail de notre impétrant du jour. Ensuite nous aurons Jesper qui vient de Danemark, Université d'Aarhus. Et puis moi-même en examinateur. Et puis enfin nous aurons le pléthorique encadrement, en commençant par Pierre et Maximilien qui prendront la parole en premier. Et puis donc les deux encadrants, ou comment on dit, co-directeurs officiels, donc François et puis Alexis. Et puis, je donnerai un petit mot de la fin et enfin, on aura le temps de délibérer tranquillement. Et toute la soutenance aura lieu, en tout cas les questions aussi en anglais, pour permettre à tout le monde de comprendre. Et donc, je donne la parole à Joachim. Bonne chance pour les 45 minutes prochaines. Merci à tous pour le silence, attention à vos téléphones et à tout à l'heure.
Hello everyone. I'm Joaquim Estopinan. Thank you all for attending the PhD Defense. PhD Defense, entitled A Predictive Approach to Determining the Joint Conservation Status of Species. Special thanks to the members of the jury for coming in person to Montpellier. So I've been conducting this research for 3 years now under the supervision of Alexis Joly, François Munoz, Pierre Bonnet and Maximilien Servageant. And I'll start now with some context and motivations. So the IUCN, or the International Union for Conservation of Nature, has established since the early 2000s a reference classification scheme to assess species extinction risk. Today, the assessments are led manually by taxon experts, and depending on the availability of data, there are up to 5 criteria assessed, spanning population size, decline, threats, and the geographic range. It results in different extinction risk categories, ranging from least concern to extinct, with the 3 threatened categories being vulnerable, endangered, and critically endangered. Now for the plants, it is often the geographic range that is being used because of the lack of availability of data for the other criteria. So this constitutes a first motivation to map species distribution. But there are, in fact, numerous motivations. So we can list, understand ecological processes and climate change impacts, guide field surveys, help to maintain ecosystem services, prevent invasive species and pathogen spread, but also optimize spatial conservation planning. So mapping species distribution influences our everyday life by orienting policies on conservation, land use, health, and climate change mitigation. So now that we've seen the motivation, how do we do it? How do we process? So a first option is to rely on the raw observation data. Here is an example for the caracal wildcat species. But as you can see, it is strongly biased by the uneven observation effort and other factors. So we need to generalize in space this information. And then a second option is to rely on expert maps. So experts from their knowledge of the species preferences and from the raw data can generalize the species range. A species range refers to the area where a species can survive and be observed. But it is a time-consuming task, and therefore it cannot be scaled up with the explosion of biodiversity data. So a third option consists in using a statistical model, SDM, to generalize in space the species distribution from the correlation between species observation and environmental covariates. And it is this third option that will be central to my work today, and will further present these models later. But first, let's further motivate the use of such models with a picture of current knowledge on species distribution and extinction risk. So to date, we know that around 80,000 vertebrate species exist, and out of these 80,000 vertebrates, we have taxonomically described, around 80% of them. And 3/4 have been assessed by the IUCN. Their extinction risk was assessed at least once. And finally, around 61% have an expert-based range map from the IUCN. Now, if we look at the plant clade, out of the around 400,000 plant species estimated to exist, we have described around 90% of them, but only 14% have been assessed by the IUCN. And finally, around 1% have an expert-based range maps. So this highlights two important knowledge shortfalls. First one being the assessment shortfall between the number of species described and the number of species assessed, which is vast for plants, but also for other taxa. In that regard, automatic methods to complement manual assessments of extinction risk can help, as we will further see in the highlights 2 and 3. And a second shortfall is the difference in the number of species described and having expert-based range maps, which is called the Wallacean shortfall. And for this shortfall especially, species distribution models can help. For instance, in the Americas, the BIEN initiative has already allowed to map 100,000 species, thanks to the Maxent model. So now the explosion of data allows new possibility to study ecological system at scale. And first, I will present the principle of species distribution models. So every mapping attempt relies on a set of geolocated observations. And we sample the environment, some environmental covariates, at the position of each observation. So, thanks to these measurements, we can now map the species observation in a new environmental space. For instance, here, rainfall, depending on rainfall and soil pH. I represented here only 2 dimensions, but this environmental space can actually be of very high dimension, as it will be the case in our further studies. Now, it is also in this space that the model generalizes the potential niche of the species. And back in the geographical space, we want to map the potential niche where the environmental requirements of the potential niche are satisfied. So we can approach this continuous potential niche by sampling regularly in space, for instance, with a global regular grid. And for each point, we test if the environmental covariates used are meeting the requirements of the environmental niche. So if it is the case, we can consider the points as potential presence for the species. Otherwise, they are deemed not suitable for the species. So now there are plenty of different model class and model properties to do that. So let's present a few of them. First, as classic environmental covariates, we can mention the bioclimatic variables, land cover, and remote sensing projects. And the environmental covariates can be sampled uniquely at the position of the observation, or a small data patch can be can fed the models in order to characterize species habitat around the observation. Now there are different model types. We can differentiate broadly traditional statistical models like GLMs from machine learning models like the maximum entropy approach or a deep learning approach that we call DeepSDM. And these kind of model are especially suited to process large volumes of data, for instance, of remote sensing data. We can also make a difference between models relying only on the presence of species, or model relying on presence and absence data, and model processing one single species at a step, or multiple species together, letting information to be shared between species. So now, ecology and species distribution modeling has entered a world of big data. But how do we define big data? So a common framework is to use 4 dimensions: volume, variety, velocity, and veracity. And the increase of biodiversity data along these 4 dimensions allows new possibilities to study ecological systems. And machine learning is especially suited to take advantage of this increase of data. An example is the PlantNet application, where a deep learning model is used to identify users, the most likely species on users' photos. And then a collaborative voting system is used to refine the identification when needed. So today, the application has enabled 1 billion identification, and around 20 million observations have been pulled on the GBIF, which is the Global Biodiversity Information Facility. So now a major concern is to take advantage of this increase of data with environmental covariates to map species distribution. So this is the overarching motivation of my thesis: improve the knowledge on threatened plant species by combining growing ecological data with new modeling techniques. So I organized this presentation around 3 central research questions. The first one being leveraging the temporal revisit of recent satellite imagery missions, thus characterizing species habitat in time, improve species distribution modeling. The second one being how can deep learning help to map certain species at both high resolution and on a global scale. And third, can deep SDMs produce species features that are informative of the extinction risk status while being flexible enough to test climate change scenarios? So here is how I organize the remainder of this presentation. First, we'll focus on the observation data that we use across our studies. Then we'll focus on the model class, the convolutional neural networks, and finally on 3 different highlights linked to each research question. And the first highlight will rely as environmental covariates on satellite image time series, and whereas the 2 others on the global rasters, as we will further see. So now the observation data. So we chose to base our modeling work on the orchid. But why? What is special about them? So it is one of the largest family of flowering plants with around 31,000 species spread worldwide. And they have always attracted the attention of both botanists and citizens for centuries. They are a threatened family, in part due to their high sensitivity to environmental changes and their biotic interactions with trees, fungi, and pollinators. And so these specificities make of orchids good indicators of the health of their ecosystem. So this is the main motivation why we chose to work with orchids, but actually we could transfer the modeling work that we have done with other taxa. And as we have already seen on the whole plant clade, we have still scarce knowledge on both their spatial range and extinction risk. So we started our experiments from a set of occurrences from Alexander Ziska and colleagues that was taxonomically filtered from the GBIF. And next, we will enrich this observation data with high dimensional environmental covariates. So it includes around 1 million occurrences of 14,000 species, and it is highly imbalanced in terms of the number of occurrences per species. So here I plotted the cumulative share of occurrences against the cumulative share of species, and the black lines indicate that the 90% of the rarest species only gather 9% of the occurrences. So this is a common bias in opportunistic biodiversity data. The occurrences date back from the last century up to 2019, and they have a global extent. As we can see also on the map of the observation effort, with two hotspots of observation, Europe and Australasia. And the orchid diversity, which is known to be higher around the tropic, is not aligned with the observation effort. Finally, what are the IUCN statuses of our orchid species? So, among the species in our dataset, among the 14,000 species, only 6% have been assessed by the IUCN. And among these 6%, half of them are threatened, either as vulnerable, endangered, or critically endangered. So this highlights, once again, the need to complement the existing IUCN information with automatic methods. So now I'll introduce the model specificities. So we chose to work with convolutional neural networks. Why? What is the rationale behind this choice? So they allow to process high-dimensional inputs and capture spatial patterns within. They are the reference model class for computer vision tasks since 2012, and common example of classification are face recognition or object detection. In ecology, we can also mention species identification, as we saw with PlantNet, or land cover mapping for planning or fire monitoring. And throughout our work, we chose the reference architecture, the Inception D3, which has 24 million parameters. Now, DeepSDM are a deep learning, we call DeepSDM a deep learning model used to characterize species habitats and model their distribution. So using a deep learning model allows to jointly process high-dimensional environmental covariates and extract critical information from multiple species at a time without overfitting on the training data. It also allows to benefit from the latest data science techniques and from a high inference capacity, as we will later see. And in our lab, a recent study has shown that convolutional neural networks can take advantage of the spatial structure of the environment. And we will see in the first highlight that they can also take advantage of the temporal variation of the habitat around species observations. So now I'll introduce some notations. So we denote as X the set of predictive features associated to each observation, and Y the set of species of our data set. So our full data set can be denoted as a subset of X times Y of pairs from an unknown probability distribution. Now our model output is an estimation of the categorical probability distribution, where eta hat of S, eta hat S of X is the conditional probability of presence of species S when the set of environmental covariates X is provided as model input. It is important to denote that this is a conditional probability of presence. So for the training procedure, we use 2 GPUs in parallel from the Shenzhen National Supercomputer, and around 70 epochs were necessary in average to reach model convergence, and it took a few days. And importantly, we used the modified cross-entropy loss function, which was designed for multi-class classification problems with strong class imbalance, as it is the case in our dataset. Finally, we use stochastic gradient descent to optimize our models. So training, shaping species distribution with a deep learning model is is still a relatively recent research avenue, and it requires to have access to important computational resources and a good understanding of the principles of deep learning to reach model convergence. So now that we've seen the training procedure, how do we independently train and evaluate our model? So to do so, we use a spatial block split of our observation. We cannot split randomly the observation of our dataset, as it is often the case in deep learning, in machine learning, because of the autocorrelation of the environmental features, which would lead to an overestimation of the model's performances. So instead, we split the geographical space with a global regular grid, and then we attribute each cell either to the training set, to the validation set, or to the test set. So in our case, we chose to attribute 90% of the cells to the training set and 5% to the validation set to regularly evaluate the model during the training, and finally 5% of the cells to report the final performance. So now, how do we effectively evaluate the model on the validation and on the test sets? So to do so, we use two metrics. First, the top K accuracy. So for a given occurrence, xi, sy, we test if, when provided with xi as input, as environmental input, if the model returns the correct label, si, among the k most likely species. So if it does so, it is a success, and ak of y takes value 1, and otherwise it is 0. So then we can compute the success rate for a set of n occurrences, all species combined, and we will reach the top K accuracy. So in this first metric is strongly representative of the performance on common species, because each occurrence has the same weight in the computation. So that is why we chose to work also with the second metric, the top K accuracy by species. And now we compute the success rate independently in the first time for each species across its own occurrences. And then we average out the species scores. So now every species has the same contribution in the metric. And as we have seen before in the observation dataset, we have a lot of rare species with few occurrences. So now this metric is representative of the performance on rare species. And together, these two metrics are suited to presence-only observation because they do not require absence data in their computation as the AUC, for instance. So now I present a first highlight. Here the objective was to highlight, to put in place a method to test the contribution of satellite image time series to the contribution of to the performance, of deep SDMs. Indeed, today, species distribution models do not often take advantage of the temporal variation of habitats, and if they do so, they often use the temporal variability of the bioclimatic variables. And now, remote sensing offers data, characterizing data on species habitats at both high resolution, high spatial resolution, and high temporal resolution. So it has long been a challenge to take advantage of such data to map species distribution. And deep learning may provide the solution to map species distribution from remote sensing data. So, to start, we had to gather satellite image time series around each observation. So we sampled image time series around each observation. So around each of our 1 million observations, we sample 12-month time series centered on each observation. So it was quite a tedious task that took me several months because I had to select the least cloudy projects, Sentinel-2 projects, for each target tile, but also process and download 100 terabytes of data, mosaicing the tiles when needed, and finally extracting the images around each observation, images of 10-meter resolution and 64 by 64 meters. So here are 4 example time series, with each time a photo of the species observed and the position. So on the top line, there are the RGB images, and on the bottom line, the infrared. And now, we'll see the consequences on the model performances. So to test the contribution of the image time series, we put in place sensitivity tests. So here in the middle, we have the original time series in chronological order, and we train a model, a reference model, with this unaltered original time series. And we also trained 3 other models with where the input time series were modified. We wanted to alter the temporal information. So a first modification consisted in randomly permuting the month of the time series and feeding the model with, for each occurrence, a series which was randomly permuted. The second modification consisted in taking the temporal average month of data and duplicate it 12 times before feeding the model. And finally, the strongest modification of the time series consisted in taking randomly a month of data and duplicated it also for each occurrence. And so we trained 4 different models with these 4 in all inputs. So now let's see the consequences on the overall performances of the model. So on the left, I plotted the top 30 accuracy, and on the right, the top 30 accuracy per species. So on the left, we are representative of the performance on common species, and on the right on rare species. And in both cases, the model with the full temporal information, so with the time series in a chronological order, performs the best, and the model having access to only 1 month of data, 1 random month of data, performs the worst. And the 2 others reach mid-range performances. So this is the first illustration of the contribution of the time series when learning species distribution. Now we also looked at the model performance in function of species rarity, or more precisely, in function of the number of occurrences per species in the training set. So on the left of the x-axis, for common species, models, all 4 models perform the best, because it is easier to model the distribution of common species, who have a lot of occurrences to learn the distribution. But for species having fewest occurrences, it is a lot harder for all models. But interestingly, the relative difference of performance between the full model and the other models, especially the model M3, is higher for rare species. So it is for species having few occurrences that having access to the full temporal information especially helps performances. Now that we thought that the same pattern would occur when looking at the model performance in function of the number of species per region, but instead it is the region diversity that seems to drive the results spatially. So here I plotted the model performance again against region diversity. And on the left of the x-axis, in polydiverse region, when there are few different habitats to differentiate, it is easier to identify the habitats corresponding to a given species. So models perform the best. But when habitats are really diverse, it is a lot harder on the right of the x-axis. And again, it is for diverse regions, in diverse regions, that having access to the full temporal information especially helps model, because the relative difference of performance between the full model and the 3 others is higher in these regions. And we can also see these results spatially. So here I plotted the same variable, but by region. And it is broadly around the tropical region that the difference of performance, with or without the full temporal information, is the higher. So these results were published in the first article in Frontiers in Plant Science. And I also had the chance to present them at 2 conferences, one in Montpellier and one in Montreal. And finally, the data and software, so especially the Deep Orchid series, are available online. So now we'll focus on a second highlight, where the objective was to map threatened species distribution at both high resolution and on a global scale. So for that, we used different environmental covariates, because we needed environmental covariates that are globally available, but also easier to handle in terms of computational costs. So we designed indicators of species diversity and extinction risk. So first, I will present the environmental covariates used in that regard. So the criteria was to be globally available, and we sampled them at 1 kilometer resolution, and the images associated to each observation are much larger than before. They measure 64 by 64 kilometers. So the first data source exploited is the bioclimatic variables from WorldClim. And on the right, I plotted the data patches associated to a given observation, always the same. So the second data source exploited were the pedological variables from soil grids. The third, the human footprint detailed rasters. Fourth, the terrestrial ecoregion of the world, here with the global map. And here is the associated patch to the same occurrence. And finally, the position was provided to the model, making the model spatially explicit, as we provide it as model input. So now the objective was to predict assemblages of the most likely species worldwide. So to do so, we train and exploit a deep SDM, as previously introduced. And for each point, we threshold the level of the relative probability of presence on species. So we have to optimize a threshold lambda on the level of the conditional probability of presence. And to optimize this threshold lambda, we use the average error control setting, also called conformal prediction. So here I plotted the accuracy when predicting if the validation species is within the predicted assemblage against the level of the threshold lambda. And with this setting, we guarantee that with our threshold, the validation species for a given validation point is within the predicted assemblage with 90% confidence. And if we give now equal weight to every species instead of every point, we guarantee that the correct species is within the predicted assemblage with 80% confidence. So this is an important point of our model, having the correct species within the predicted assemblage with 97% confidence at the occurrence level and 80% confidence at the species level. So now we use this setting to predict the assemblages of the most likely species worldwide on a global regular grid of 30 arcsecond resolution. So it resulted on more than 200 million points, and we will see later the results. But first, we define or we used indicators that are based on every point on the predicted assemblages and on extension risk information. So the first indicator is a common biodiversity metric, the Shannon index, which is representative of species diversity and evenness in the predicted assemblages. The second indicator developed is the most critical extinction risk stages within the predicted assemblages. And the third indicator is the proportion of species with a given extinction risk status in the assemblage. So for the two latest indicators, we can either consider all the species in the predicted assemblage, or we can restrain their definition only to IUCN assessed species. So if we consider all the species within the assemblage, we need to complement the extinction risk information with predicted statuses. And for that, we use the state-of-the-art method, the IUCNN method, to complement the extinction risk information. So first, we'll see the most critical extinction risk status, but not based on our predicted assemblages. Instead, we'll use the range maps available on the IUCN red list. So here is the result. And as we can see, there are only few common species, apparently, that have range data. And the spatial coverage is quite scarce. And now if we base this indicator not on the range data available on the IUCN anymore, but on our predicted assemblages for the same species, we obtain a larger spatial coverage and globally higher indicator levels. But still, the indicator levels seems underestimated in some regions, for instance, in South America, since it is a precautionary indicator. So now, if we consider all the species in the predicted assemblage and not only the IUCN assessed species, we obtain a different map with higher levels globally and also new patterns revealed. So now let's look at the Shannon index indicator. So here we can see that globally the regions known to have a high diversity are respected. But however, there are still regions, like for instance Colombia, where the diversity seems to be underestimated. So it could be due to the observation data, but also to the modeling choices made in our method. And now let's focus on the last indicator, the proportion of threatened species. So here we consider together vulnerable, endangered, and critically endangered species, only for the IUCN assessed species in the predicted assemblages. And now if we complement once again the extinction risk information with all the species in the assemblages, we obtain a different map with higher levels globally and new patterns. And we can do the same for each threatened category independently. So here for the endangered species only, first for the IUCN assessed species, and now for all the species endangered in our predicted assemblages. And finally, for the critically endangered species, so here considering only the IUCN assessed species, and now all the species critically endangered in our assemblages, so completing the extinction risk information with the predicted statuses. Each time, levels are higher and new patterns are revealed. Now these maps have actually kilometer scale resolution. So to appreciate the patterns, we also developed an interactive website where we can zoom in the patterns. And here, for instance, in India, the Western Ghats are predicted to host critically endangered species when completing the information with the predicted statuses from IUCNN. Also, we can, another valorization option of such indicators is to intersect spatially their value with other variables. So here in Sumatra, we hashed in blue the current protected areas, and also on the top left, the elevation profile of the island. And we can see that the proportion of critically endangered species is predicted to follow the mountain chain. So it provides a new information to orient next conservation measures. And finally, we computed statistics, zonal statistic by country. So here I plotted the average threat level by country against the average Shannon index value by country. So we can see, for instance, that in Madagascar, the predicted assemblages contain, in average, 60% of threatened orchids. And we highlighted the countries with the highest threat proportion and the highest Shannon index value. And Southeast Asian countries are among both the most threatened countries, but also the most diverse countries. So these works with other analysis have been submitted to ecological informatics. And once again, data and software are available online. And the method is being reused in other contexts in 2 European projects. So now we'll focus on the third highlight of this presentation, which extends aspects explored previously. That is deep SDM range prediction and IUCN status prediction. So now our objective was to design a method to classify extinction risk, but a method allowing to explore bioclimatic projections. So that is what stands out from the state-of-the-art methods, the capacity to project the extinction risk prediction in the future along the bioclimatic variables. So how do we process? So first, we validate the method in the present with the same occurrences and environmental variables, global rasters, as before. So the first step consists in using deep SDM as a dimension reduction algorithm. And for each occurrence, we obtain a feature of reduced dimension which captures the environmental information present in the global rasters. But now we want to aggregate such information that is for now at the occurrence level, at the species level. So for that we use summary statistics to obtain such information aggregated on the species level. And finally, on the third step, we train a classifier, so a random forest classifier, on the already assessed species to hopefully learn a good function mapping the species features with the extinction risk statuses. So we validated this method on the present data, and we achieved similar results than the state-of-the-art methods. So now, in the future, we do not rely on the observation data anymore, but on 2 dispersal scenarios, either considering null dispersal or unlimited dispersal for the species. And also we replace the bioclimatic variables with projections. So projections following a business as usual socioeconomic scenario. And we consider 4 different time periods. And the process is then the same, using the same 3 steps, but now with the deep SDM trained with present data and the extinction risk classifier also trained with present data. So now the trends, the statuses obtained with this method are hard to analyze locally. So what we have done is to aggregate them by broad region or along a gradient. And it is easier to analyze the projections by such a method, by such region. So here, for instance, I mapped the species, the number of threatened species across time periods. And for each time period, I map the number of threatened species in function of the species' average elevation. And we can see that the number of predicted species is predicted to increase at mid-range elevation and also in the lowlands. Hopefully you can see it. So this method was published in Ecography, sorry, not published, submitted to Ecography, and the data and software are once again available online. So now let's synthesize the results. First, we have shown that the habitat phenology, so the variation in the habitats that may or may not occur in species habitat across the seasons that are captured in image time series, improve species distribution modeling. And it does so especially for hard predictions, so on rare species or in diverse regions. And the associated remote sensing dataset is available online. Second, deep SDMs contribute to closing knowledge gaps about threatened species distributions. And for that, we have developed global indicators at kilometer scale resolution that enable multi-scale risk analysis. And the interactive maps are once again available online. And third, we have developed a method where DeepSDM provide flexible features to classify the extension risk of species, and also a method allowing to follow bioclimatic scenarios. And with this method, we have shown that levels are increasing globally, but with alarming levels in the tropics, in the lowlands, and at mid-range altitudes. So now, we believe that the limits and perspectives to this work are numerous. So first, there is still a trade-off between models' accuracy and interpretability. Interpretability being our capacity to understand and explain predictions. And for instance, we could have further quantify the uncertainty linked to our predictions at the assemblage level or at the extension risk level. And it is actually a very active research area, explainable AI, so we could reuse some methods from the literature to further explain, interpret our predictions. And we could also reuse methods from causal inference. Second important point, crucial point, is to have more dialogue with conservation actors, especially involving them early in the modeling process in order to let them take part in the modeling decisions. And I think it is also important to analyze the results together, especially from a spatial point of view for conservation. And it is also important to develop flexible tools in order to adapt the results to real case studies. So third, on the modeling side, we could use new architectures like vision transformers to better capture the spatiotemporal preferences of spaces. And in that regard, the GeoLifeClay challenge is fostering modeling developments by providing a ready-to-use dataset. And methodological advances also allow to compensate for bias in opportunistic biodiversity data, for instance, the sampling bias. And finally, at the level of environmental covariates, so first, for remote sensing data, it is a rich data source, but it is costly to handle at very large scale. So its use should depend on the modeling objective, on the target resolution, and on the extent of interest. And some proximal features on species preferences are hard to collect at high scale. So for instance, for orchids, we know that the distribution of mycorrhizal fungi is very important, but it is hard, if not impossible, to collect at large scale. But however, numerous other data opportunities exist, like essential biodiversity variables, species co-occurrences, and species traits, or deforestation projections. So, thank you, that's all.
Merci Joachim, on va attendre quelques secondes le temps que certains quittent la salle. So thank you. So the first one is Catherine. You can start with Joachim.
Okay, do I need a microphone?
Yes.
So I would just like to start by complimenting you on a really nice, both a thesis and a very clear presentation that you just gave us.
Thank you.
Yeah, and in particular, what I appreciated about your thesis was just the scope of questions that you tried to answer and the different types of methods you tried to apply to look at these questions. That was one thing. And secondly, though I guess your background isn't ecology and evolution, you made a nice attempt to try to put everything in this sort of framework, which I also realize can be quite challenging and really quite useful. And then finally, I really appreciated this sort of attempt to provide sort of useful or interesting guidance in terms of conservation and IUCN red listing. And so I have a series of questions, which of course are not perfectly on the page in front of me, and I need my glasses to find them. But maybe I'll just start sort of in the middle of my questions, and or even at the end of your thesis. And one of the things, well, I'll just ask you first, so what is the main reason that species are listed?
Sorry, what is the main?
What is the main reason that species are listed or classified as endangered or threatened?
So I think it's to possibly orient conservation, or to maybe to prevent some, I don't know, maybe constrictions to harm the habitats if they are identified of these very species. But yeah, in practice, I guess that red listing a species does not always have a practical implementation of the measures. But yeah, and maybe also red listing species can orient policies on the different objectives that are made by countries. But yeah, if the question is about the practical consequences of red listing a species, I must admit that I don't know them all. But hopefully for some species, maybe the most charismatic ones, some measures are taken in order to prevent their extinction.
Good. So if I'm in a certain place and I have a list of species, how do I decide that species A or species B, whichever they are, should actually be considered as threatened or as vulnerable, or we shouldn't worry about this species? How does the IUCN make this decision to say this species should be on the list at this category?
Okay. So there's actually 5 criteria. I have a slide on that somewhere. Oh, I went too far. Yeah, this one. So yeah, there are 5 different criteria spanning different variables. So the first one span the population size reduction in time, and the second one, so the criteria B, is the one that I highlighted here because it is the most used for plants. For the large majority of plants, we use these criteria. So where we enter the criteria with 2 main variables, the extent of occurrence and the area of occupancy. So the extent of occurrence is the convex hull spanning all the observation, and the area of occupancy is the area of the cells occupied by the occurrences. And so we confront these 2 first variables against the threshold, and after that we also have to satisfy 2 other sub-criteria among 3. But in practice, the sub-criteria B is often assumed, and the criteria A is estimated, yes, more or less. But yeah, for the plants, it is a criteria that is most as used. There are 3 other criteria, C, D, E, but not that often used, I guess.
Yeah, so I would agree with that. And then when I think about when you take all your unknown data and estimate it, or try to build these estimators, well, let's start with your known data. In some sense, the dependent variable, so the listing, is based on size. Yeah. And then your thing that's explaining the listing is also size. Yeah. So in some sense, you have the same variable on both sides of a prediction. Okay. How does this work?
Yeah, if I understand well, is why do we include special variables in the input of our models, since it is also the target in an extent? But yeah, I think that we have to include spatial variables in the extension risk classifier, because since the reference is constructed that way, so it is actually the variables that explain the most the actual statuses. So I don't think, if I understood well, I don't think that there's some circularity. But yeah, definitely there's some kind of we find something at the output. The actual statuses are depending strongly on the spatial distribution.
Yeah, yeah. So could you just take a map of weighted endemism and get the same map as you got based on your fancy models? Just to be difficult?
It is a good question, yeah. I don't have tested the contribution of the different features, but it would be very interesting to test the contribution of, and maybe, yeah, such map would explain quite a large part of the variability in the results. So it would be an interesting test to run, but I can't say more about that. Yeah, definitely.
Just one more question. If you had one plant trait or a couple plant traits other than just area that might be useful to predict their extinction risk, what would those traits be? Which traits would you wish you had to do this on something than just area or just ranges?
Okay. For orchids? Or
for orchids, yeah.
Yeah.
Sorry, I should have been specific.
No problem.
Thank you.
It's a good question. Maybe, so knowing their life form is important first, I guess, if they are epiphyte or not, but also maybe having the pollinator information would be interesting to cross it, but I don't know if even if the pollinators information, I'm not sure we could actually use it for predicting the extinction risk statuses. Maybe we could. There's actually a lot of data on birds, so maybe it could be useful to cross it, but I'm not sure. So I would say life form first. Maybe, yeah, preferences in terms of temperature. I'm not sure.
Yeah, it would be interesting. I think in mammals or animals, it's often body size, right?
Yeah.
But I don't know what the equivalent is in orchids, I have to admit. Maybe somebody else does. Good, and then you have all these patterns of risk in the end. Did you compare those to, say, other taxonomic groups? So could you just have studied birds and mammals and gotten the right answer, or did you actually need to also study orchids?
Okay, so that is a good question. So I think the technique, the model, is transferable to other taxa, but with a condition also. Is that for species the dispersal is pretty much null at a small time scale. So I guess we could transfer the method to other taxa, and if we have compared it to other maps from animals, I have looked at other methods more to see what exists, but I do not have compared the outputs. So it was more, I looked more on the methodological side, I guess.
Yeah. No, your thesis is definitely methodological, so of course I'm going to ask the questions that are more ecological, and then you'll get the methods questions, I'm sure, later. So now I'd like to, I'm sorry, where were those notes? I'm a mess. So in your second chapter, you keep pointing out mountains and highlighting mountains, and then you have something called the mountain gradient diversity theory, which was a new one for me. So can you tell me what that is, or what you meant? I think this was in chapter 2.
The mountain gradient diversity
theory.
It was when we talk about the Sumatra case study.
Yeah, or maybe it was in chapter 2. You have this.
Oh, sorry.
And you call it. You have this name.
I'm not sure I could tell a lot about Monte gradient diversity, but maybe, yeah, there is. So don't worry about the slide. I'm not choosing it. Yeah, in the mountains, then there are some areas where I read that that are most suited for having more species speciation and a lot of different species, because they are quite remote from anthropogenic threats. So they are like refugees for now, but with the climate change, they are beginning to be impacted by the changes. But I'm not sure it's the answer that you expected.
Yeah, I think you called it the elevational diversity gradient theory.
Sorry.
And I was really quite curious. So when we think about elevational gradients per se, how does richness tend to vary across a gradient, an elevational gradient? Do you know?
Normally, the richness is higher at mid-range elevations, but it's poor at high elevations.
Yeah,
right?
Tends to be.
Yeah.
Mm-hmm. And the lowest elevation depends a bit on whether it's a wet bottom or a dry bottom. But usually mid-elevations or low elevations has the highest, but not high elevations. And so people always talk about mountains as having such high diversity. Why is that? Is that a function of alpha diversity or of beta diversity?
In terms of...
just number of species.
Okay.
Is it because they're high in alpha? Is this high richness because of how many species are in a given place? Or is it because overall mountains, there's a big beta diversity driving that richness?
I would say both, but it's the easy answer, because alpha diversity is on the number of species, and beta on more on the habitat level. So I guess that since there is a big gradient of elevation, there's also a lot of variation at the habitat level. So I'm not sure.
So I think if you just took 1 hectare of tropical lowland forest and 1 hectare of even mid-elevation mountain forest, you would have more species in the hectare of lowland forest.
More?
Probably. Oh. But if you were to look across hectares that were separated by 1 kilometer, you would have more species in the highland forest, just because of the high beta diversity. Okay. So when we talk about mountains and their high richness, it's not always just local diversity in a given spot. It's because there's such high beta diversity and change, right? And when you think about scale, and you talk a lot about scale in your thesis, you touch on that very nicely. And so if you have these big grid cells in mountains, they're going to contain a huge elevational range, and for that reason, a ton of species compared to the same size in the lowlands. Okay. So, yeah, it's just an interesting thing, because everybody correctly thinks that mountains are very important for biodiversity, and they are. But any given spot on the mountain isn't necessarily so crazy diverse. Okay. It's just because of the beta diversity and the history of that beta diversity. Anyway, so this elevation diversity gradient theory didn't mean something so specific for you, which is good, because I'd never heard of it. I even looked it up to see if I'd somehow missed this theory. I'm just giving you a hard time. Okay, and so then one of the other questions I had about going back to the conservation, you did this really nice sort of analysis of which other species could be at risk, right? Did you take that any further? So did you ask what characteristics these species had, or maybe you didn't have other characteristics? So you have this long list of, I would call them, we used to call this latent risk, or things that potentially could be at risk or are at risk, they just haven't been identified as a risk. It's like the dark diversity of risk or something. Do you know were there characteristics of those species that you could identify?
Unfortunately, we did not go that far. It would have been really interesting to look at the clusters, try to understand a bit more why some species are threatened or not. But so that's one of the perspective, I guess, further analyze the results. yeah, I don't have
Just
elements.
curious. It's actually super cool. It would be nice to know. And then another sort of gets back to a theoretical question. These aren't in any good order. You talked about the potential niche, which I thought was interesting. So can you tell me the difference between the realized niche and the fundamental niche, which are the first original concepts? And then what is the potential niche?
Okay, so the fundamental niche is the niche where the species could occur without considering biotic interactions, if I'm right. And when we consider the competition and other mechanisms, it becomes the realized niche. And the potential niche, it was a concept introduced a lot after. And I don't actually remember exactly what it involves more, but there are other constraints that make it kind of smaller or more constrained than only considering competition. But I'm sorry, I don't remember exactly the elements.
So I mean, there's a huge discussion about what niche is being modeled by distribution, and so there's probably, I have my own opinion, but you probably don't want to assume that my opinion is right. But when you model a species distribution, you're taking information from where it really occurs. You really believe it's there, right? And so which of those niches might that represent? Why is that a potential niche and not, say, a realized niche?
Yeah, it's a hard question. And I don't have the exact answer. It's a common, convenient way to answer also is to say that we try to model a continuous function of species preferences instead of a niche, or at least not that the reality is not that binary, but it's a convenient answer, of course. And I had another element. Also, since in our model we include the position in the input, I read that it is quite different. So from models considering only bioclimatic conditions, for instance, and it makes the interpretation of the output different too. And so I'd like to say that we map the potential geographic range of the species, since we already include the position in input. So
and you actually call it a species distribution model.
It's a spatially explicit distribution model.
Nice. Good. And then in your first chapter, you did this huge effort to get all this temporal data, which I really appreciated. And then you didn't show us, but in the thesis, you compared that also with the climate variables, and sort of sadly, the climate variables were better predictors, which was frustrating since you got all of this fine scale data. And so do you really believe then that the climate variables are always going to be better predictors, or when might this effort for the fine scale sort of data sort of pay off?
Yeah, it's a good question. So first element, I guess, is that since we evaluated our model on a global scale, so I'm not an expert on that side, but I guess that on the global scale, it is always the bioclimatic variables that will explain more species distribution. But the interest of having remote sensing data, I think, appears at smaller scale. So for instance, if we have evaluated the model more on more local tasks, I think here we would have very different results. So I would say, it depends on the scale of evaluation and also on the target resolution, of course. We cannot, if we want an output map of high resolution, we have to rely on high resolution inputs. So I think it's strongly application dependent, but on the global scale, of course, bioclimatic variables will always have explain a large part of the variability, I guess.
So I guess you could have done it with some European orchids, right?
Yeah.
And then maybe it would have shown to be more important. Okay, so I will let someone else start asking you questions now. Thank you.
Thank you.
Cool. Yeah. So, well, first off, thank you for the very nice presentation. It was really enjoyable also to read your thesis. I just actually only realized today that your PhD was entirely within a computer science department. I kind of assumed that you had also some biology background because I had reviewed computer science theses before, but they were like harder to read. This one was really like a pleasure to read as a computational biologist, but really with a biology background, so really nice. I have a number of questions that are mostly methodological. They mostly come from my curiosity of like trying to understand. I guess I haven't really checked your codes, which I think you very nicely made all available. I wanted to understand a bit more about the architecture of the first model, like that used in chapter 3, I think, and then so for species distributions, and then the second model where you do these species assemblages. So in the first model, I wasn't quite sure if you actually used pseudo absences for training or not, because like you said, like then the model evaluation did not use pseudo absences.
Yeah, we never used pseudo absences actually, because we don't need them as in the evaluation process. We only rely, at least we don't need them in the evaluation that we use. But of course, it would be useful to have absences. But yeah, in the I don't know if I can find it. Yeah, it's about the metrics that we use. So the top K and top K accuracy, they don't rely on absences. We only look at if we can predict the correct label, so the correct from the validation set within the most likely species predicted by the models. So we don't confront, as it would be the case in the IUC, the false positive rate or false negative rates. So it is in that sense that we don't need pseudo absences, at least to evaluate the model.
But was your loss function based on these metrics, or were these metrics used like afterwards to evaluate the model?
Okay, no, the loss function is a cross entropy loss function that is modified to give a stronger emphasis on rare species.
And that also was based only on presence-only data, like without
absence data. There is actually label smoothing involved in the inception differential architecture. So the objective is to repredict the loss function with penalize all the species except the correct label. But there's a lot of regularization techniques with label smoothing that tend to soften the penalization, I guess. But yeah, the loss function is already with the correct label. Yeah.
Okay. Yeah. And also, in this LADAM function, like, does that function add any parameter or hyperparameters, or is
it relies on the prior, on the number of occurrences per species, and there is a rewriting scheme that appears at so you can set it after a given number of epochs, and it gives a stronger emphasis on the rare species. So I have a graphic for that somewhere. I try to find it. No. Yeah, here. So here is the two validation loss. So it's on the second chapter. First, we want to train a deep SDM before predicting the assemblages of the most likely species. And so I differentiate the calibration model to first evaluate the performances, and the final model where we consider all the data set, all the data together. But what is interesting about the loss is here. At the epoch 65 is when the rewriting takes place. And we can see that the rewriting impacts the performance on common species on the left. because the performance drops for the common species, because here it is a top K accuracy, not by species, so every occurrence has the same weight, and it improves the performance on rare species. So it is a consequence of the reweighting in LDAM, which give a stronger emphasis to rare species after the reweighting. Yeah.
And it's more efficient than just like super sampling or giving different instance weights?
Yeah, it's a good question. We do not have confronted different methods, but yeah, it has a very strong. The LDAM is a lot cited and a lot used, so we just assumed that it was a good manner to.
Yeah, just curious. Okay. Cool. And then in the other model where you do the prediction of the assemblages, then so there you have a softmax at the last as the output function.
Yeah.
And then then you set a threshold to decide like which species are actually going to be in the assemblage and which not. But then so the softmax function or like the model is trained to give basically low but equal probabilities in places where you expect to see a lot of species, which I don't know, I found a bit strange, like because then in a species poor assemblage, then softmax will like kind of push for like high probabilities for few species
for
and
the
very low
first
for
one.
others.
And, yeah.
So does that like affect the accuracy that you would expect to see in species-rich assemblages? Partly because softmax has this tendency to make like probabilities either very large or very small. So it's like usually it has a hard time like finding stuff in between.
Yeah, it's a very good question. And if the model grants a too high probability to the first species, it would be quite harmful in these techniques, since we do not rely only on the first one, but take a whole assemblage. But when we looked at the distribution of the probabilities and the output of the model, it was actually quite not that peaked. So I guess there are a lot to do at the level on the level of calibration, and there's plenty of more things that we could do, but I don't know, at least our model did not peak that much, so it was quite balanced, the probabilities as output. So there's definitely more work to do at on this level, but yeah.
Cool. Yeah, thanks. Again, I'm trying to see. Yeah, I guess like, so, yes, where you use these predictions of extinction risks, I think it was really nice to extend, I mean, I was involved partly in this IUCNL model, so it was interesting to see that from a different, like, kind of path you reach like similar accuracy, and also that like you can then extend your model with like climate predictions in the future. So that was really interesting to see. I was a bit surprised not to see that like the high elevation species are, I mean, I would expect them to be the most endangered in the future, but you seem to find that it's more at mid elevation that the extinction risks increase the most. Do you have an explanation for that?
Yeah, it's a good question. We did not analyze a lot the result at these levels, but I think also you mentioned, for instance, on the distribution of threatened species across latitudes, that Madagascar could play a big role. And so maybe there's some local explanation of such patterns, and that definitely would be worth investing more time. But no, I don't really have an explanation for now why we don't observe more threatened species at high elevation, higher.
Cool, yeah, thanks. So in that model, like the species, so you have these dispersal scenarios, and then like species will just no longer occur in a place.
Yeah.
So they will become extinct if they have no other place to go, basically.
Yeah,
right?
that was the hypothesis, yeah. When we use the dispersal scenario, so the two extreme scenarios, either null dispersal. So in this scenario, the species can be repredicted only where they were once observed. It is in this scenario that we observe extinction, or what we consider extinction. In the unlimited dispersal scenario, there are never extinction. Always, a species has always a point where its probability is high enough to be above the threshold lambda. But yeah, when so in the first case with null dispersal, there were for some species, for any point, they were repredicted. So where they were once observed above with a conditional probability of presence higher than threshold lambda. So it was in these cases that we assumed extinction.
Yeah. Yeah, so one thing that could be added to the framework is that, I mean, these extinction risks are defined as probabilities of extinction. So if you're critically endangered, you're 50% likely to go extinct in 10 years or something like this.
With the first criteria?
Yeah, I mean, these are like usually the definitions, right, of these labels. And then I guess like when doing these experiments in the future, you could account for this probability of stochastic extinction based on the labels. So if you project the labels through time in the future, you could also predict the probability that the species actually goes extinct even before climate like actually kicks them out of their own of the range. But they could just go extinct because of stochastic fluctuations.
Yeah,
as predicted by the labels themselves. Yeah.
Yeah, it would be worth interest.
But I mean, it would make the map even more sad, I think so.
Yeah, there's plenty of other variables to account for.
Yeah. Yeah. So thanks. I think this is all for now. Thank you all. Thank you very much. Yeah.
Thank you.
Next one is Jesper.
Thank you. All right, Joachim, I want to start by thanking you for a very nice thesis. I really enjoyed reading it. You've been covering a huge amount of different methods, and you've been processing huge amounts of data. So it's really a big undertaking, and you should clap yourself on your shoulders for having done that.
Thank you.
Yeah, so I think I would start by following up a little bit on Daniel's question about this threat, because you, in one of your maps, you show that the threat status is relatively predicted to be relatively low in Europe, and I would actually, I was a little bit surprised by that, because I would think that Europe is among the areas in the world where you have the highest population density, and you have a lot of agriculture and a lot of forestry. So I was a little bit like surprised by that map. Can you comment on that? Why do you think you don't get some more threat in that region?
Actually, I think it's also related to our method. So because it depends on the predicted assemblages. So in Europe, I guess with our threshold on the level of probabilities, there are lots less species predicted than on diverse regions, and that's maybe a first reason why the threat levels appear lower. And also, concretely, so here we have a global map, so the color scale is maybe pushed up by the really threatened regions, so kind of making the region with lower levels a little bit more invisible. So maybe it's also a problem of appreciating the patterns globally, is not maybe giving enough importance to such region in Europe.
Right. So because when you look on your maps, it looks like there's a big overlap between where you predict the highest threat to be and the Shannon diversity. So it's where you have the most species, you also generally find the most threat level, basically, right?
Yeah.
So it could be funny to make a map where you like calibrate for that or account for that, so that you could.
Yeah, definitely. Yeah. It would be interesting to include.
All right. And then continuing in the IUCN red list criteria, I wanted to ask you, because you showed the figure when Catherine asked you about the criteria.
Yeah.
And it seems like your model actually is based mostly on the geographical information.
Mm-hmm. Yeah.
Can you comment a little bit about that? Because you have 4 other criteria that you can red list as a species on, and when you use only the geographical one, don't you risk that you are missing a lot of species that would fall for the other criteria in the IUCN red listing process?
Okay. Yeah. So it's a good question, and for plants, I don't have the percentage in mind, but I think that's something like at least more than the half of the assessments are made by the criteria B.
Okay.
If it's not 80%, I should have checked that.
Okay.
So the reference is constructed that way. So since the reference is built with the criteria B on the geographic range, it is often the best to try to approximate to optimize with to target with automatic methods, since the reference is built like that. But yeah, if species have been red listed with other criteria, it would be more difficult to spot, to learn. The correspondence between the threats and the species features in our model, since we mostly rely on geographic information, but still there are also other inputs to our dataset, the anthropogenic threats and pedological bioclimatic variables. But yeah,
Okay.
definitely.
Thanks. Then to something different. At one point you write that, let me see if I can find it actually, on page 93. So you write, bioclimatic variables outperform Sentinel-2 image time series in the determining global orchid distribution.
Mm-hmm.
Static variables also achieve better test performance than satellite data with positional information likely to make a large contribution. So why use the Sentinel-2 data? Why not just use the bioclimatic variables? I guess it's very cumbersome to use the Sentinel data. Yeah.
Yeah, so I think it all depends on the modeling objective once again. So on the global scale, using the bioclimatic variables, at least for our evaluation, was shown to be sufficient, even better. But I think with other metrics and more local assessments, the temporal time series, so satellite data would be shown, would be highlighted a lot more important. So it all depends on the. It's a good question, but on a global scale, yeah, definitely bioclimatic variables appeared enough and even better. And for the static variables, in fact, we include a lot of information, and especially position, which is a strong driver here of to generalize the distribution of species. So I think this is why we reached so good performances.
Okay. I actually also had another passage that I wanted to read aloud. It's on page 120. So you say, however, it is essential to remember that no single mechanism fully explains a given pattern, that interscale dependencies and local historical events strongly influence biodiversity, and that no pattern is exempt from variation and exceptions. And then further down you write, remote sensing is a natural perspective for improvement. The inclusion of biological functional traits of orchids is another exciting perspective, as well as mycorrhizal fungi or pollinator distribution. So I wanted to ask you, does that mean that remote sensing is never going to give a perfect or near perfect models, because we need these other kinds of informations that we cannot really derive from the remote sensing data?
Yeah, I think we will always need field data to complement the remote sensing data, because there are interactions that we cannot account with remote sensing data. So I think they are bringing complementary information, and so definitely remote sensing brings a lot, but it cannot account for I don't know, mycorrhizal fungi once again for orchids at least, and so maybe that's why multimodal models taking advantage of different data sources appear really promising to try to extract the valuable information first from each different data source, each modality, and after try to mix them in order to take the best. But I don't think that we could only rely on remote sensing data, or depends maybe on the taxa. For some taxa, I guess big trees, it would be nice, but it's better to cross information, I guess.
That was actually touching a little bit upon my next question. So do you think that there are some species groups that are easier to predict than others? And if so, why would you think that?
So it's a good question. I think it depends on the species preferences, on the species dependencies. So if if the species can rely on I don't know, in patterns, on biological elements that can be easily spotted with remote sensing data, definitely it would be easier. I don't know, maybe interaction with bigger species once again, something like that. If there are strong markers of their presence. So in that sense, I think that their species are a lot easier to map than others, even if in my case I only looked at orchids, which are not easy to spot, of course, but that's why we work on a habitat level, not on the species level.
But I guess that you could also maybe say that some species, so coming back to the niche terminology, you could say that some species are probably, they have a very broad niche, and other species have a very narrow niche. So maybe you could expect that would make a difference in terms of how easy it is to actually make accurate prediction maps, because some species would be able to thrive in a broad range of different environments, right?
Yeah, if the niche is well differentiated, it would be easier.
Right. I think I just have one more question. If I can find it in my. Where did it go? Sorry. Okay. Yeah. So there's just one more thing, because now, as you may know already, now I am actually doing something that is a little bit similar to what you're doing. Namely, I'm trying to predict things that doesn't have a true answer, and that's actually what you are doing. So because you are predicting the status, the IUCN threat status of orchids, and they are assessed, and that means that a given category, for example, endangered, it's not like the finite answer of that it's actually endangered. It's the best assessment given the data currently available. But so what challenges do you see in that you are trying to predict something that is actually not necessarily true?
So it's a good question. But in our cases, we consider the IUCN assessment as the truth, the reference. But maybe it could give ideas to complement the assessment. But so because to date it is the criteria that are most legitimate, and I think it's a good thing because we rely on the same set of criteria for all species, and it gives a strong weight to the red list. But maybe including some complementary approach would be interesting. But yeah, it's a hard problem. We have to rely, I guess, on some reference data, but definitely as they are false positive and false negative in our cases, there must be also false assessment kind of in the red list.
I think I don't know if it's possible, but it could be worth considering to try to account for that in the loss function. That it could be equally true if it predicts vulnerable or critically endangered.
Yeah.
Even though it should predict endangered.
That is why we tend to consider more binary predictions, or only threatened or not, because it is a lot easier, especially for the categories near threatened and vulnerable that are the harder to predict because of their definition. So near threatened is basically near the official threshold, but not quite. So it is a quite large definition. So yeah, definitely involving some uncertainty and is at this level, and allowing to predict, for instance, the category above or on top would be interesting. And actually, there's also methodological work that so here is not involved, but in IUCNN, I think that you can quantify the uncertainty linked to the predictions. So definitely, it's useful.
Okay, thanks. I could continue forever, but I'll stop now.
Thank you.
Thank you, Jesper. So I will continue a series of questions. My first one is just, you started with that, with Catherine. It's about abundance. So for plants, it's not so easy. So what kind of adaptation do you suggest to model abundance with many zeros, with the kind of data we can get for animals or over taxa?
So how could we evaluate, estimate abundance from such model? So it's a good question. So we have to rely on different observation data if we want to predict abundance, and I'm not sure how many, how available they are. So there are some database of plots where it could be interesting to rely on. So
yes, plot is a good example.
Yeah.
So what is your strategy to model cover or plant cover in plots, for instance?
So I think it would involve a different evaluation scheme. So since with plot data, we would have the knowledge of which species are here or not. We would try to predict the I don't know. Here we use the top K accuracy because we don't have the list of species that are present at a given position. But with the plot data, it would allow new evaluation models. So I don't have other. I didn't have a think about that more precisely.
Do you think it's better to have a kind of two-stage process? I mean, first predicting occurrences as you did, and then trying to predict abundance where species are present, or you think it's better to have a kind of single model predicting?
Yeah, using a single model makes a lot of sense, and I think it all depends on the available data, because here we used like a two-steps approach, at least for the predicted assemblages, because we could not do something else. But yeah, predicting directly with plot data, the assemblages would be nice.
My second question is about less sophisticated models. I was a bit concerned about the lack of comparative or comparison you provided in your papers. Did you try more basic models like species-specific, not considering all species together? And did you try more basic machine learning models, all of those things, as benchmarks?
It's a good question. And no, in my case, I didn't do such comparison. I relied on the work done by colleagues before, where they have shown that this class of model was reaching similar or better results for species. So it was done in France and in the US with data sets on these two countries. But no, so on my case, I relied on such results, but I did not explore and test other alternative models. But definitely it would be interesting.
Yes, for instance, on a graph like this, it would be great to know where you are when using just a basic GLM or machine learning.
Yeah,
just
definitely. Yeah. Mm-hmm.
Okay, I just missed that in your thesis, but not so easy in 3 years. So thank you for that. So now you're nice supervisors.
Thank you.
On va faire dans l'ordre.
Très bien, merci. Thank you, Joachim, for this presentation. So I would like to introduce my equation by big thanks to the work that you did. And it was not an easy context, even if if this work has been built based on previous research results obtained by colleagues and previous PhD students. So it was not easy for you to manage a context in which you have a co-supervising team with so many different point of view and expectation on your work. So it was a really great period for us, I think, to work with you. You did a big effort in order to aggregate training data in the first years, and it was not guaranteed to reach such a diversity of results in 3 years. And I think that we can be impressed by the capacity and the investment that you did in the transfer of your research results. So you show that some of these results are now available through a web interface, and you mentioned only in one line in your presentation that your method is now used in the context of projects that will take advantage of this work, and it's not so common to have such transfer during the period of the PhD. So congratulations for that. So coming back to the question that I have, so there is some kind of, we could say, a big run by spatial agencies that promote and increase the accuracy of the satellite data that we can use for many different purposes. And can you let us know if you think that an increase of the resolution of the satellite data could increase necessarily the performance of the models that are based on that data? So coming back to the first experiment.
Okay. So thank you for this question. So I think it depends always on the mapping scale, but on a regional scale, definitely having access to better resolution of spatial, better spatial resolution of satellite data would be interesting to provide output maps orienting choices after. And depending on as input to the models, I think it would be an interesting test to run. It would definitely depend on the taxa models, but and on the performance of the different models in function of the resolution of the satellite data would be a nice test to run. And I hope so. I think so, that it would help having access to a high resolution, but yeah, it has to depend on the taxa, I guess.
And for example, in the graphs that you have in your bag, so do you think that model based on Sentinel could reach the best result obtained by the use of the other modality, or there is some kind of limitation that could lock the capacity of this model to take advantage of higher resolution?
With the same evaluation scheme at the global scale, I'm not sure it would lead to better results, having access to a higher spatial resolution. Yeah, but once again, a lot because it's at global scale.
Yeah. Okay. There is something that you investigate during your study is the impact of the GPS resolution, the spatial resolution in the occurrence that you use for the training. So several orchid species are known by a very low number of occurrences. These occurrences are quite old, so they are based on a records that were based in a context where we could not have access to GPS. And so, do you think that in the future we could find a way to increase the benefit of these old occurrences, for which we will probably never increase the spatial resolution? But do you see some potential strategies that could allow to still continue to take advantage of these quite old occurrences in order to improve a predictive model?
Okay. So I think we could take advantage of older occurrences, but distinctly from more recent occurrences where we could have a good match between predictors and the observation, because it would be the most qualifying information. But having a model that combines the older occurrences, maybe linked with features that are at larger scale, would be interesting, and combine the learning on these older occurrences with the newest occurrences, where we have a good confidence of the match between the predicted data and the occurrences. Would be maybe a first strategy to take advantage of the older occurrences. That definitely would have something to bring.
Thank you, Joachim. And I will conclude with a last question. So you have worked on the most large number of taxa as possible within this family based on the available actual occurrence and a volume of at least a few hundred occurrences per species. So you work on 14,000 plant species in a family that count around 30,000. So for the last highlight that you share with us, what do you think could be the tendency for the 50 plant species that are not represented in this study and that cannot be represented due to the low volume of data available for them? So can you elaborate a little bit more about these species that could not be covered and took into account for this type of study?
Okay. So the first point, I think that the species for which that are not included in this data set, have less data. So they have less. They are less represented on the GBIF or on the biodiversity portals in general. So I think it's an indicator that they are quite hard to detect, or maybe hard to observe, or I don't know if we can directly consider that they are rare species, but they are not. At least they are not observed for now. So it's maybe as a precautionary approach, we could consider them as more likely to be threatened. But so I'm not sure, but I would consider that they are more likely to be threatened as more rare, since they have not been observed a lot for now. but maybe it's just because of the sampling effort. So it's quite hard to know what is it. We can also consider that the dataset is large enough and the proportion would be the same for the unobserved species. But yeah, it's an open question, definitely.
Yeah, an open question, probably interesting to discuss with a specialist of this family.
Yeah.
Exactly. So we will see if this can be followed by a future direction. So thank you very much again, Joaquim.
Thank you.
Thank you, Pierre. I want to thank you for all this year we have together to work on because human qualities and scientific qualities were very important, and the relationship was very good with that. And also we have a side project that you did not mention about the Western Ghats. And in this side project also you show the ability to see together the biological issues, ecological issues, and the statistical methodological issues. So I think it's very important to be able to integrate both sides in order to do some sensible indicators.
Thank you.
But I have a few questions. And one is, we have already discussed, is could we consider that the orchids are kind of umbrella taxa? And in which case, can we use directly the information about the presence of threatened orchids as an indicator of necessity of conservation action in many kind of habitat?
Okay. So there's a theory about bioindicators or species that can be considered as good indicators of the health of the ecosystem. And so one of the important points is to be sensitive to the environmental changes that occur. So if the species reacts to changes in the environment, and so orchids have this property that they are sensitive to the changes in their habitat, and so their presence in that sense, they can be considered as umbrella species because they indicate how well the habitats around them, where they are observed, is doing. And so there are other elements, other important elements to be considered as a good indicator of the environment. And I don't remember all of them, to be honest, but yeah, I also relied on a thesis done especially on orchids, where the author was interested in that question, and he was quite positive. So I trust the results and cite him a lot. But I would be interested in knowing more of the links between both.
Yes, and also maybe we already discussed about this point. You have very different strategies, ecological strategies among orchids. And maybe we have enough information. For instance, I was looking at the database about epiphytic orchids, and terrestrial orchids which are in forests, terrestrial orchids which are in open areas are very different kind of ecology. So maybe one of the perspectives would be to integrate in the tool, in the mapping tools, in the classification tool, the percentage and identifying, for instance, in a tropical forest, if you have a lot of endangered epiphytic orchids, of course you should not cut the forest, for instance, something like that.
Yeah,
another important point we already discussed before was can we only consider global indicators of IUCN status when you need to have some local scale conservation strategies? What I mean is that in France, for instance, you have an orchid society, and they were not happy with global indicators. They did their own red list for France only. And this is the red list which is used, in fact, by people who want to manage habitat.
Okay.
So my question is, can we have a way to tune the spatial scale? And for instance, species which are very common in Scandinavia are quite endangered in France, only in the mountains, for instance. And then we need to acknowledge that.
Yeah, so there's a methodology to regionally assess the species on the red list. So to adapt the global assessments at the regional scale. So I think for France, they have used such methodology to assess at the national scale, but still with the red list. And so the question was about how to bring together the assessments at national level and at global level. If I okay.
Maybe there is not the same need, in fact. Maybe at the local scale, you need to consider that species are very sensitive to the local scale environmental change. On a global scale, maybe, for instance, you have a lot of rules about the trade of orchids, because you have a lot of people collecting orchids in tropical forests, and we need to have a strong regulation of that. So maybe the indicators of global scale are quite relevant for this kind of policies, you see?
Yeah, and considering, so for instance here, orchids at different scale can result in different priorities. So it could be quite complicable. Hence here we try to develop multi-scale indicators, but it's quite, it's a hard work, a hard task. So it's not perfect far from here, but I think it's the objective, try to have indicators that can scale up and down. So in order to reconcile priorities both at global and local scale.
Yes.
But yeah, it's definitely an open question because the local priorities would not.
Yes, it's basically the point of view of managers, in fact, which is important to acknowledge, because when you use some conservation indicators, you need also to be clear for to what objective you can use it.
Yeah, and that's it.
And also, last question is that so we can have a status for a species, but we have also the possibility with your modeling approach to have a status for occurrence, meaning that, for instance, you can have a variation of UCN status within a species according to different occurrence places. That's it?
Yeah, I think it is linked also. We could link this point with the quite recent now red list of ecosystems, where so we try to qualify the extinction. It's not extinction risk; it's risk to collapse of ecosystems. And so yeah, maybe species is not the only organizational level that should be considered to prioritize the conservation measures. So looking at the occurrences of a given species or other levels would bring complementary information. And yeah, I think of course for a given species the threats can vary from a population to another. So it's important also to say that it's already
yes, I mean, for instance, if it has a high ranking in some place and a low ranking in another place, maybe it can show that it is more sensitive to any change in the habitat.
Yeah, it happens frequently to be regionally threatened, but globally not threatened.
Okay, I will stop question, but I just want to thank you again for this work, and I hope that we could continue to use this data set and to work on this kind of topics together in the future, and maybe in Grenoble.
Thank you.
Thank you.
Merci. I only have one question, but first I would like to thank you for your presentation, for your well-written manuscript, for these 3 years, and for all the scientific discussions we've had during working hours and outside working hours.
Thank you.
My question is about your background. You come from an engineering school, mostly focused on computer science, and then you joined the Zenith team, composed of researchers in computer science, applying machine learning models and statistical models in ecology, mostly in ecology. At the end of your PhD, you found a postdoc position with Wilfried Thuillet, and now you are joining a team of ecologists applying these models in ecology. And my question is, at the end of these 3 years, what do you favor? The biological phenomenon, the reasoning about ecology that you can bring together with mathematical models, or the thinking about these mathematical models?
Yeah, it's a good question. But what I identified, I don't know if you will be happy with this answer, but what I've identified to like is precisely the intersection of both. So that was one of the criteria, both to choose the PhD, the project initially, and also for what comes, it was to have kind of one foot on the two fields. So it's quite hard to choose, or at least I don't want to choose between both. And I don't want to. I like to do methodological work also. So I think it would be maybe frustrated to personally, in my case, to work only on the interpretation of the methods without deeping in the how it works. But yeah, it's a good question. Definitely, I have interest. That's why I chose these subjects. Mm-hmm. And yeah, you mentioned that. So now I will be in a laboratory of ecology, because it was one of my not concern, but I assured that it was also a lot methodological work. So I think I'm still in both.
Thanks.
Thank you. So thanks a lot, Joachim. So it was a real pleasure to supervise your PhD during those 3 years. I think it was like a hard context, as a lot of first said, but there is big data is hard. I mean, coding, debugging, it's very hard stuff. And you also spend a lot in investing in the ecological domain, trying to have the right phrasing, as it was said. And even in ecology, you are between conservation and ecology. And some of the questions were related to that. So the IUCN status, I don't like them, but for you, it's a reference. So, and these are other multidisciplinary stuff, I think, that could be discussed. So yeah, you are between a lot of domains and stuff in that. So maybe I will just bother you with one just technical question that is like rephrasing the one of Jesper, because I'm sure you can provide a more complete answer. So regarding the softmax, so imagine you have one specific geolocation where you have a lot of presence-only data without bias. And so here you have a lot of presence data of all the species. And you have another location data, leisure location point, where in the same environment, so exactly the same, but with very few data, so like the 2 dominant species or something like that. So where do you think the softmax is going to peak? And so what does it say somehow about the convergence of the softmax?
Okay, so it's indeed a very technical one. I'm not sure, actually. I would say it would peak while there are few candidates. It is easier for the model to peak when there are only few candidates. But when there are a lot of different species in the first point, I presume if the model is well calibrated, the form, the distribution of the probabilities would be more balanced.
And if you have an infinity of points?
That's true.
So, thank you, I think. Provide some highlight on that. And I think that's all. I let the president.
On va devoir délibérer, donc vous demandez quelques minutes de sortir de la salle avant de le féliciter. Merci. Celui-ci. Voilà, donc à l'unanimité des 3 rapporteurs et puis de tes superviseurs, donc on te décerne le titre de docteur de l'Université de Montpellier. Comme tu sais, il n'y a pas de mention particulière, mais on te félicite tous pour tout ce travail. On a relevé, tu verras dans le rapport, donc une grande qualité de présentation, d'illustration, beaucoup de travail, une grande clarté. et surtout voilà, une grande capacité à avoir fait ce côté interdisciplinaire, écologie et puis sciences informatiques. Donc encore bravo, félicitations, et puis bonne chance pour la suite.
Merci beaucoup. J'ai beaucoup aimé travailler à Montpellier avec cette équipe. C'était vraiment 3 années de recherche intéressantes dans un environnement privilégié, je pense, donc autant sur le plan professionnel que personnel, c'était vraiment très agréable. And now, I think I have something to read at the moment. And also, do I read it now? Yes. No, okay. So, in the presence of my peers, with the completion of my doctorate in computer science, in my quest for knowledge, I have carried out demanding research, demonstrated intellectual rigor, ethical reflection, and respect for the principles of research integrity. As I pursue my professional career, whatever my chosen field, I pledge to the greatest of my ability to continue to maintain integrity in my relationship to knowledge, in my methods, and in my results.
Voilà, au nom de tout le jury, on te félicite and thank you very much for those coming from abroad. Thank you very much for coming to Montpellier in presence.
Thank you to everyone. And so now we'll have you're all invited to a first po. I don't know how would you say that. So just outside the amphitheater. You'll see, you can't miss it. And after that, there's a second one at the Brock Cafe. So it's next to the plants garden, yeah, to make sense with the with the thesis. And it's from 7 p.m. So you're all warmly invited.